So my name is Lais. I'm a research associate at the Lab of Translational Cancer Immunology and Immunotherapy and King's College London here in the UK. And today I'm going to talk a bit about my work that I did during my PhD, where I basically purified chondroitin sulfate from a shrimp and tested its anti-tumor and anti-angiogenic activities. First of all, what is chondroitin sulfate? So, chondroitin sulfate is a type of glycosaminoglycans or GAGs for short. And GAGs are carbohydrates that present in each structure repeated units composed of an acid, uranic acid, and an exosamine. And depending on the type of the different units that are combined together, you can then therefore have different types of GAGs, which can be heparin, heparin sulfate, chondroitin, dimethan, or keratin sulfates. And on the top of that, you can also have additions of different positions of sulfate groups in the molecule, also Foucault's branches. And as you can imagine, if you play around with all those different combinations, you can have quite a big, so it make GAGs being like a very heterogenic molecule. So they have a very diversity structures. And because of that, it makes them very unique in the context of they can bind to wild type of different biological proteins, including coagulation factors. In fact, the majority of GAGs are known to have anticoagulant activities. As you saw, heparin is well known as an anticoagulant agent, is widely used. GAGs, different GAGs are also known to bind to cytokines, chemokines, growth factors like FGF, different proteins of this family, metalloproteinases, and a variety of different cell receptors. And this is interesting because if you think about it, this is just a few of the proteins that GAGs can actually bind and interact with. And I'm not just talking here about like cell to cell interaction, but like cell to matrix as well. Cell that includes immune cells, different immune cells, different tumor cells, and also GAGs can play a role in the infection of different pathogens. So if you think about it, they play a very crucial roles when we talk about normal physiology and also in the pathology itself, and especially in cancer, because different types of GAGs have been found to perform signalization within the tumor environment. So because of that, for the past years, GAGs has been studied, and mainly for the past maybe a couple decades, those studies have been focused on finding new GAGs from other animals, and the main focus being now in marine animals, because the main reason is that apparently GAGs from these animals, comparing to GAGs found in mammalian animals, present very unique structures. And that's one of the reasons why the lab where I did my PhD and my master's, we focus on the shrimp because this whole research was performed in Brazil, and it's a very common type of shrimp that we have in our coast in Brazil. So our lab managed to characterize 4 different types of GAGs extracted just from this species of shrimp, the Lithopeneus vanameae. But we are going to focus on the last one, which was the subject of my PhD, was a chondroitin sulfate. And that's what we're going to discuss here a little bit. So this chondroitin sulfate, after we did the extraction and the purification, we managed to do the structural characterization by using magnetic resonance. So, and we found a very interesting, unique structure for this compound, where it presents 3O sulfation and the glucuronic acid of almost 60%. And why this is important? Because for chondroitin sulfate, this is quite rare. So you not find very often molecules, especially from mammalians, that have this particular structural feature. So that also, together with the fact that we saw some in vitro and in vivo anti-inflammatory activities, and also that the fact that this compound actually presented a very interesting anticoagulant activity, mainly mediated by the inhibition of thrombin, that rounds us like to pursue more studies, further studies with this molecule. And we mainly thought, well, if this compound actually is able to inhibit so strongly thrombin, maybe might play other biological roles within other biological subsets apart from inflammation. So we were interested to see the main activity of this compound on the inhibition of thrombin. And that's because thrombin is a very interesting protease in the sense that thrombin is known, so high levels of thrombin circulating in the blood is known to actually able to activate a specific receptor called PAL1, which is a protease activated receptor. So it's a specific receptor for thrombin that a lot of cells, endothelial cells, and especially tumor cells has in the surface, which once activated by thrombin, it promotes different signalizations favoring the tumor growth, like proliferation, invasion, and angiogenesis, just to name a few. And in other words, high levels of thrombin can cause a more pro-tumor environment. So in that sense, we start to look in our compound to see if, because of its capacity of inhibited thrombin, we could therefore be maybe in a direct way before this compound could show anti-tumor activities. So that was the thing that we mainly did in this study. Here I'm only going to describe the in vitro work. We also performed the in vivo part of the study, but we're writing the paper right now. But we can, if any one of you have an interest in on it, we can contact me and we can discuss further. So the first thing we did, we want to know if in the presence of the SCS, the cells would present any harm. It could harm the cells, like present any kind of toxicity. And for this entire study, we used the murine melanoma model, where we used the B16F10 melanoma cells. As you can see, we performed cell viability assays by Alamar Blue, and all the concentrations tested of the compound, and also in heparin, which throughout the study we use as a control. We can see that it didn't affect at all any viability of the cells. We also perform flow cytometry, and we can see here clearly that neither heparin or the CS promote any kind of apoptosis or induce cell death, really. So that makes a question. So can, for example, can maybe this compound be inducing some kind of anti-tumor activity by other mechanisms, by inhibiting thrombin and then indirectly mediating this effect? So we pursued the studies, and then we did an in vitro long-term proliferation assay, where we incubate the cells on the top of agarose coating plates, and we left there for 10 days. And what you can see, those dark dots that you see are actually the colonies, because melanoma are known to form colonies. And you see very clearly here that when you treat those cells in different concentrations of the compound, you actually see a very significant decrease in the colony formation. So that was quite interesting because this is a long-term experiment, and the compound showed this effect, both anchorage independent and dependent, which just made us more curious, and then we decided to perform also an invasion and immigration assay, and we were trying to understand which mechanism this assay would be able to perform in the anti-tumor effect here, and how potentially. So what we see as well is that in the wound healing assay, we could see that in the presence of the compound in both and all the concentrations tested, we see that even after 48 hours, the compounds were able to decrease the invasion of the tumor cells and compare it to the control. And all those effects so far have seemed to be dosage dependent. So in the highest concentration that we use, which was 100 micrograms per ml, we found the better effect here. The same was also observed for the transwell migration assay, where when we incubated the cells in the presence of SCS, we saw a very important decrease in the migration of those cells, which is a very important mechanism in cancer. As if you think about an invasion, and which is a crucial mechanism that the tumor cells had to use to actually to produce metastasis, for example. And we came to a hypothesis here. Well, one of the things that is well known that we already know because of the studies in the literature is actually the fact that GAGs are able to bind. Like I said, GAGs have unique structures. They are big molecules, they are chargedly negative, so they very easily bind to different biological proteins. And is well known that the GAGs are able to bind to cell surface proteoglycans like CSPG4, for example, in melanoma cells. GAGs are well known to also bind quite strongly to different growth receptors and all the, like I said before, the extracellular matrix proteins. And the hypothesis here is that maybe the SCS was able to bind to those proteins. They are so important for promoting migration and invasion. And therefore, those proteins were not able to bind to their specific receptors and then promote all those cellular mechanisms. So this is one of the hypotheses here, at least so far, for these results. And when we had all those results, we started to get more curious, and then we decided to basically see if the presence of the SCS in the medium could somehow modulate the expression of genes that are important for melanoma progression. So what we see here is we did the PCR, we did RT-PCR and we chose genes that were relating to melanoma progression. And we wanted to see if there was any kind of modulation in the presence of the compounds with the melanoma cells. And as we can see here, pretty much all the genes that we study suffer some kind of modulation that expression, especially the CX43, which is a connexin, is very important for adhesion and also important to the contact of cell to cell. For example, promote angiogenesis, for example. And we also saw important modulation in NF kappa beta and via GFA, which NF kappa beta is a gene that encodes the NF kappa beta, which is a transcription factor, which orchestrates all the secretion of cytokines that in the tumor microenvironment play a very important role in controlling and improving the spread of the cancer, basically, and the growth. And in the case of the VEGF-A, it encodes the growth factor, the vascular factor VEGF, which is the main factor that it helps to promote angiogenesis, which is important for tumor metastasis. But unfortunately, we could not see a significant modulation in the expression of for the AP2 alpha, which is a transcription factor that is responsible to modulate the expression of PAR1, which was the thing that we're mainly interested here. And we could also not see any expression levels of PAR1 here when we performed those experiments, nor even in the controls, which we think is due to the incubation period of the cells, because it was only 48 hours. And maybe we thought that later we could have to leave the cells longer, so we could see significant levels of this gene to actually show up in the expression. But what I can say here, when we perform the same setting of experiments, the same PCR with the same subsets of genes in the in vivo, using melanoma tumor samples and lung samples from the animals, we not only not just were able to see the expression of PAL1, but we also saw that in mice treated with SCS, we had a huge decrease in the expression level of this gene, which was quite, yeah, it was quite encouraging for us to continue with the studies. We also saw the genes that are involved in the secretion of the interleukins, like the PAF receptor, FGF, and also when we talk about whole and MAP kinase genes, which are crucially in the proliferation and tumor growth. So yeah, those results were interesting for us because they kind of corroborated what we've seen with other dysfunctional assays in the immigration assays, invasion, and the long-term proliferation. So that kind of guided us a little bit towards to a possible explanation what one of the possible mechanisms that SCS was promoting these anti-tumor activities. We also decided to perform quantification of melanin production by the melanoma cells. And the reason why we've done that is because in the literature, melanin by melanoma cells is actually correlated with the aggressiveness of the cells. So the more the cells become more aggressive, the more they will produce melanin in an indirect response. And it was quite interesting because we collected the supernatant of the cells with the different treatments, different concentrations of the SCS compound. And we saw that in the highest concentration of the SCS and heparin as well, we saw a decrease in the melanin levels. And this is quite interesting because you can somehow use melanin production as an indirect way of seeing whether the cell is more aggressive or not, and that it relies on the fact that the melanin production, the melanin synthesis by the melanoma cells, it actually falls in the pathway which requires p38 phosphorylation and then native activation. And this pathway is one of the pathways that culminates in the activation of the MAP kinase gene as well. And this is important because this gene is, like I said before, and this kinase is related to proliferation, adhesion, and invasion. So it's quite crucial within this tumor environment to promote tumor progression. So it was kind of interesting to see this correlation between all the results and with the gene expression results, the proliferation results, and now this kind of confirmation with the melanin production. We also quantify the levels of TNF-alpha secreted by melanoma cells in vitro. And even though is TNF-alpha, we think that is a type of cytokine that actually fights the tumor and it helps to kill the tumor. It actually was shown that depending on the tumor and it depending on the stage of this tumor as well, the type of the cells, you can actually see that TNF-alpha is actually help the progression of the tumor. So, for example, TNF-alpha was found to actually upregulate metalloproteinase levels in melanoma cells, which therefore facilitate the invasion and therefore metastasis of this tumor. So what we saw here is that the cells was actually producing less TNF-alpha in the presence of SCS and both heparin as well, which once again was corroborating with previous results that we've seen for this. And lastly, because angiogenesis is so important for the tumor microenvironment, for the success of the tumor invasion, we decided to see whether SCS would prevent the tubular formation of endothelial cells. So it's a very simple experiment where you want to see structures that resemble the formation of vessels, so which is the tubular formation. And what we saw here is that actually in the highest concentration of SCS tested, the compound was able to inhibit almost 100% of the tubular formation. So we were really impressed with these results. And even though we've performed the gene expression PCR to see the gene expression, we using melanoma cells, and we saw that there was a decrease in VAGF levels, we could kind of extrapolate for here and saying like, maybe this compound is promoting not only maybe modulating the expression of important genes for this process, but we also know for a fact that the majority of GAGs are known, especially heparin. There's loads of studies showing the heparin can not only bind but inhibit different growth factors, especially VAGF, and therefore by competition again stopping this protein to bind to its receptor and therefore promote its mechanism to promote angiogenesis here in this case. And once again, it's interesting to see that you could think, oh, maybe the presence of SCS was so much that promote toxicity of the cells and you kill the cells, and then that's why you couldn't see the tubular formation. But when we performed the cell viability assay, we also saw that the presence of the compounds actually did not promote any kind of toxicity on the cells, which points out that this mechanism, which why SCS is promoting an anti-angiogenic effect, it might be for a different type of modulation, and that's what was being describing here so far. And as a conclusion for this study, what I would like always to say is that I think is this new and unique structure of this chondroitin sulfate actually help us to understand better the relation between functionality and structure, and also this could be a useful kind of molecule to be used as a model to develop new molecules that can act as with anti-cancer potentials. And with that in mind, I think it's important to know as well that this study needs to be continued as well. There are the gaps, we are aware of them. When there are more answers, we still need to see whether the gene expression modulation correlates with proteins production as well. So we are continue with this study, even though I'm not in that lab anymore, but I know them the team is seeking with that. And I also would like to say that all the in vivo studies that we've done confirm everything that we've seen in vitro. So both in the inhibited the tumor growth in mice, but also in decreasing angiogenesis as well. So as I said, it's an ongoing publication. So we are actually writing the paper right now. But if anyone has any curiosity about, we can of course talk later about this. Yeah, I would like to thank all my lab amazing team where I work in Brazil, and where I did my PhD, my master's, my scientific initiation. I also want to thank for my PhD supervisor here in the UK at the University of Liverpool as well, to all the programs of financial incentive in Brazil for me to perform this work, and to the place that I work now, which is this King College, for allowing me to be here participating with you. So thank you for the opportunity.